Okay, good day, everyone. I will be continuing the discussion for direct immunoagglutination. So, hoping this is not your bedtime story. Okay. So, hoping this is your teleserye ng tutok ba kayo mo pa. Okay. Sige. So, another test for direct immunoagglutination test would be your febrile agglutinins. Okay. So, these are agglutinins that are produced against fever-producing bacteria. So example of these diseases, you have typhoid fever, paratyphoid fever, brucellosis, tularemia, and typhus. Example of this test is your Weil-Felix na test. So Weil-Felix test detects heterophile antibodies produced by patients with rickettsial disease. So if you can still recall sa rickettsia, so it's a bacteria that is vector-borne. No? So it is transmitted through bites of ticks, mites, or fleas. So there's a common antigen between your rickettsia and some strains of your Proteus. So you have there OX19 and OX2, which are strains of your Proteus vulgaris, and your OXK, which is a strain of your Proteus mirabilis. So it's a titer of 64 to 320 is suggestive of this disease, and there's a fourfold increase of the titer between paired sera gani is already diagnostic. So your OX19 will react with the antibodies against typhus group, while your OXK will react with the antibodies against scrub typhus na group, and your OX2 will react with the antibodies against spotted fever na group. So you have the slide method. So you make serial dilution gap of the serum, 1:20 until 1:320. You add it with your antigen, and then you observe agglutination in 3 minutes. And you also have the tube method. So this time, a tube method gani, longer gina siya incubation time. Okay, you have there 52 degrees Celsius for 18 hours. So ang false positive ani na test could be the other fever-producing bacteria po, no? Such as the disease of brucellosis. Next is the serologic test of your syphilis. So again, syphilis causative agent is Treponema pallidum. So you have two serological tests. You have treponemal test, so which detects the specific treponemal antibody of class IgG, and your non-treponemal test. So non-treponemal gani, meaning it's detecting your reagin. So reagin is an IgM antibody directed against phospholipids, no? So if ever there is cell damage sa lawas, possible pod maka-release or maka-produce ang body og reagin. Okay? So in syphilis, it is detected in the serum during the first and the secondary stage of syphilis, and it will already be detected in the CSF if nag-late stage na siya of syphilis or tertiary. Okay? And the reagin will actually increase, no? Or it can be detected 4 to 5 weeks of the infection, and it will still be increasing during the secondary stage, and then will decrease siya in your tertiary stage. Okay? So what are examples of your non-treponemal test? You have your Colmer, Klein, Hinton, Mazzini, CAN, RPR, and VDRL. So as mentioned, dili siya specific for syphilis. So you have the biologic false positives. You have your malaria, leprosy, typhus, infectious mononucleosis, lupus, atypical pneumonia, trypanosomal infections, infectious hepatitis. And you also have physiologic false positives. Okay, you have pregnancy, drug addicts, or more than 70 years old. So not necessarily nanay infection ang patient. Okay? So one of the tests that we will be discussing is your VDRL. So for sure na hanas na kayo mo ani, no? So VDRL uses serum or CSF. So first thing that you have to do, so when you talk about VDRL, no, the meaning is Venereal Disease Research Laboratory. So dire siya first na discover. Okay? So first step is to inactivate the serum. So at again, 56 degrees Celsius for 30 minutes. The reagent used here, so the antigen, the main source of your antigen is your cardiolipin. So by the name itself, lipin, it's a phospholipid. Okay? So this is extracted from a normal beef heart. Okay? And then you have your lecithin and cholesterol, which is to increase the sensitivity of your test. So the lecithin will neutralize the anti-complementary activity of your cardiolipin, while your cholesterol will enhance the complement fixing capacity of your cardiolipin with your reagent. So you have there the qualitative test of your VDRL. So again, using concave slides. Okay. So you have there, you added 0.05 ml of patient serum plus 1 drop of your VDRL antigen. So just a simple recall, unsa ang bevel gigamit? Gauge 18. I rather unsa ang needle gigamit, no? The gauge of the needle. So again, so wrong na siya, the bevel is actually cut off, no? Para uniform ang size of the drop. So tanggalon, i-cut ninyo ang bevel, then you're going to use the needle na gauge 18, dispensing 1 drop. So that is equivalent to 1/60 ml. Okay? So after that, you rotate it, mechanical rotator at 180 RPM for 4 minutes. Then you read it, no, under the microscope in the LPO. So you have the reactive, weakly reactive, and non-reactive na reading. Okay, so if it's reactive gani, medium to large clumps with few or no free antigens. Okay, can be seen. Okay, if you can recall man good, if your antigen is mixed with saline, there is uniform size of rod particles. Okay, if ever na ay reagin, so mo clump siya. Okay, so therefore you need to take note not only on the size of the clumps, but also on the free antigens. Okay. When you say weakly reactive, small clumps are seen with few to moderate. So magadaghan ang free antigens. So non-reactive, no clumps are seen. Okay. Next, you have your quantitative test for VDRL using ceramic ringed slides. Okay. So quanti na gani, meaning you have titers na. So from 1:1 until 1:32. Okay. So this time you're using NSS. So na NSS ani. So wrong ang bevel, no, but it's the gauge of the needle. So gauge of the needle used for NSS is 23 with the volume dispensing of 1/100 ml. Okay. Then you also have antigen gihapon. Okay, so the gauge of the needle used is this time it's 19. Okay. And it will dispense 1/75 ml. Okay, ang 1 drop. So 1 drop ra gihapon. Okay. So you can still recall kung anong different ang gauge of the needle na gigamit for quali and quantitative. Okay? So recall, kay different man ang volume of your serum used, di ba? Sige. So you have there 1 drop of antigen is added to 0.04 ml of dilutions, rotated for 4 minutes at 180 RPM. So same ra with your qualitative. So here, significant titer is 8. So if the titer is more than or equal to 8, meaning the patient has active syphilis. If it's less than 8, latent or dormant ang syphilis niya, previous treatment, or late syphilis na siya. Okay. So condition reaction is an example of so you have there, kanino, mga clumps. So moderate, so few clumps naman na siya with few to moderate na mga free antigens. So this will be your weakly reactive na reaction. Okay. Okay, so just a simple recall sa inyong quantitative, sa inyong syphilis. Okay. So inyong first three reaction circle, okay, this is undiluted na serum. Okay. So you just added 0.04 ml in your first reaction circle, giving a 1:1 na dilution. Then you added 0.02 ml here sa inyong second reaction circle with NSS na 0.02. So meaning 2 drops. No, kay kaning na gauge will give you, 1 drop will give you 0.01 ml man. Okay? And then 0.01 ml na siya for your third reaction circle. So with 3 drops of your NSS. Okay, so that's why you have a dilution na 1:4. So while your fourth to sixth reaction circles, you are going to use the diluted na serum. So giunsa man pag-get ang diluted. So 0.01 ml of the serum plus 0.07 ml of your NSS. Okay? So from that, you're going to put 0.04 ml in the fourth reaction circle, giving you a 1:8 na dilution since wala siya NSS gibutang here. Okay? While your fifth reaction circle, you have to add 2 drops. You have to add 0.02 ml of your diluted. Okay? So since your diluted has a dilution of 1:8, so your final dilution is already 1:16. Okay? And then in your last reaction circle, you're going to add 0.01 ml of the diluted. Okay? And 3 drops of your NSS. And then everything, all the reaction circles, you're going to add 1 drop of your VDRL antigen. So equivalent to 0.01 ml of your antigen. Okay? So dili na siya i-appeal sa inyong pag-get sa inyong dilution, ang inyong antigen. The next, you have your RPR. So RPR is using still VDRL antigen with choline chloride and charcoal particles. Okay, so ang purpose ng choline chloride, no need to heat the serum to inactivate the non-specific inhibitors. Okay, so siya na daan mo inactivate. Charcoal particles to view it macroscopically. Okay, so this time it is not suitable for CSF. Okay, so you're using plastic coated na cards with 14 to 18 millimeter sizes of circles or your reaction circles. So your procedure, you add 0.05 ml of serum plus 1 drop of your RPR antigen, which is equivalent to 1/60 ml. Rotated at 100 RPM for 8 minutes. Okay. So interpretation this time is, di na ka mo read og weakly reactive, no? So you just read reactive versus non-reactive. So kato di ang quantitative. So what you will consider as the titer is would be the reciprocal of the highest dilution that showed a reactive na result, no? Dili siya weakly reactive na result. So kani, for this one, example of your RPR, no? So kaning naaragali siya solid na black di siya considered as clump, no, but it's because of your antigen magud nagigamit no with charcoal. So solid ragay siya sa tunga, that is negative. Pero na ni clump sa kilid, okay? That will be positive. Okay? So basic from this one, ang titer niya is after more 1:64 man siya here. I know this will be 1:4 kani siya. 1:4, 1:32. Okay. So the titer, since the last one here is the titer for this one is 32. So kaning second na reaction circle. The second group of reaction circles, no? So this is 32. So this was used semi-quantitatively na siya. Okay, pero if ever nag-qualitative naman na RPR, so katura, reactive or non-reactive. Okay? So next, no? So that will be all for your direct immune. So kanimo hapsay ra ko og other test, no? You have your direct non-immune. So the name itself, non-immune gani, meaning the antibodies are not the ones involved in the agglutination process. So example is your viral hemagglutination. So the interpretation of the test is based on your sedimentation pattern. Okay? So imuhang viral hemagglutination is basically detecting heme agglutinins, okay, against certain viruses. Okay? So you have there's mga results possible. Okay? So you have dark red smooth button at the bottom of the microtiter well. This is a negative result. While if there are cells spreading at the well's bottom with jagged pattern or irregular edges, that will be your positive na reaction. Okay? So this time man good, ang nakakause sa agglutination are your viruses, no? So you have there, if negative siya, you have there the RBCs lang, no? So compact button ra siya, smooth ra siya. Pero once mo diffuse na galing na siya, okay, jagged patterns, irregular na ang edges because of your viruses, no? The hemagglutinins of the viruses. So that would be a positive reaction. Okay? So kani, this is just how the test is done. Okay? So namoy 96, no? Ka micro titer na well plates. Then you add your viruses with your serum. Okay? Sa inyong dilution. So this is RBCs. So dili dahil siya the RBCs as well as your viruses. Okay? So again, ang positive kay katong jagged pattern. So if ever mo compact ragali ang button, that is negative. Okay? So more of this next week na ninyo na meeting. Okay? Another direct non-immune is your phytohemagglutinins or your lectins. So which are derived from plants. Okay? So non-immune siya kay dili man gikan sa body ang antibodies. Okay? It's not the body producing the antibodies, but from plants. So you have your phytohemagglutenin L, which means leukoagglutinin, and your phytohemagglutenin erythroagglutinin. So your E. Okay? So kanay mo hang leukoagglutinin is also mitogenic. Okay? You also have your anti-H lectin, which is extracted from Ulex europaeus. Okay, na plant. So which will agglutinate your O cells. No? And then lectin from Dolichos biflorus, which will agglutinate your A1 cells. Okay? So direct non-immune gyapon na sila. Then you have your passive or indirect immune agglutination. So again, if ever passive gani, the antigen is not normally found on the surface of their surfaces. Okay? So for example, na ipabutang pa nimo ang antigen, no? You have your latex, gelatin, and silicates. Could also be RBCs, no? So with a size of 0.8 to 7 micrometers, which will give consistency, uniformity, and stability of the reaction. Okay, so mo na gina-use siya ang passive agglutination, no? Okay, uniform ang reaction, stable pagid siya. So latex are stable, inexpensive, and has no known cross-reactivity with other antibodies. So polysaccharide antigens are easily absorbed to RBC surfaces. So IgG was naturally absorbed to the surface of polystyrene latex particles. So kana bitong usual nato na ginadu sa lab. So for example, katong RF, 'di ba? So, gi-absorb ang inyuhang IgG to your latex. Okay? So, kani ang principle niya no, of your latex. So, either you're looking for the antigen or your antibody. Okay? So, if you're looking for your antibodies, so you have there your carriers. No, kaning mga bigger circles. And then you have your antigens. Okay? Then, inyo siya ibutang sa inyuhang particles. So, meaning i-coat nimo siya with the antigen and then add it with your positive na patient sample na kay agglutination. It could also be you're looking for the antigen. Unknown nimo ang antigen. So, you have there the carriers. I-absorb nimo siya ang antibodies. Okay? So, therefore, if naa ang antigen sa patient, mukha siya og agglutination. Okay? Next, a type of your passive latex agglutination wherein you're looking for the antigens and not the antibodies. So you call that your reverse passive latex agglutination. Okay? So this may be used to detect group B Streptococcus, Staphylococcus aureus, rotavirus, and Cryptococcus neoformans. It can also be used in measurement of therapeutic drug, hormones, haptoglobin, and ceruloplasmin. So in reverse passive latex agglutination, your rheumatoid factor reacts with any IgG, leading to a false positive results. So therefore, para to remove the interference no, of cross-reactivity, so usually gina-use ang monoclonal antibodies. Okay? Another type of agglutination would be your agglutination inhibition. So it's a competition between particulate and soluble antigens for limited antibody binding sites. So haptans are complex with proteins which is attached to the carrier particle. So haptans in themselves, if sila-sila ra, they are non-immunogenic. Okay? But it may create new epitopes when paired with a carrier. So therefore, kailangan siya additional para mo bigger siya size, no? At the same time na para kakause siya og agglutination. Okay? So ang naturally occurring na haptans, ang pinaka-common is your catechol, no, in your poison ivy. So here, sample is mixed with enough antibody against the targeted hapten. Indicator particles contain the same hapten to be measured in the patient. So for example, na gina-use ani would be your drugs, no, your cocaine and your heroin determination. Then you have your heme agglutination inhibition. So RBCs this time are your indicator particles. So this detects antibodies against rubella, influenza, para-influenza, your RSV, a respiratory syncytial virus, mumps, measles, hepatitis B virus, herpes, and adenoviruses. So your RBCs have the viral receptors. So therefore, if the patient has the antibody, it inhibits the agglutination. So again, ang imuhang positive ani is inhibition of agglutination. So walay agglutination. Okay? So this is an example no, of your agglutination inhibition. Okay? So you have there antibodies no, with their ang reagent, and then you have your patient sample. So you're looking for the antigen. You're going to incubate. So if ever, here is a positive result, kaning left side. Okay? So if ever the patient has the antigen, mo attach siya sa antibody. Okay? And this time, you're going to add your antigen-coated latex particles. Okay? So since if positive ang result, na-coat naman, na-attach naman ni muhang antigen, no? Unknown antigen to your antibody, walay mo agglutinate sa latex-coated na particle. So therefore, same rin siya og epitope with your unknown. Okay? So same ra gipangita. That's why nag-compete sila. But if ever negative ang patient, no, wala siya antigen. So pag-add ni mo og latex, coated particles, kay ni agglutinate siya with the antibodies, no? Na imong gipon. So therefore, ang negative test is your agglutination. Okay? So kanilang. Okay? So this is just ang pinaka simple na explanation with your agglutination inhibition. There is competition. So next is your antiglobulin mediated agglutination. Okay? Or you also known as your Coombs test. Okay? So this detects your non-agglutinating antibody. Again, is your IgG. So the component used is an antibody against human globulin that is made in animals or hybridoma technique. So talking about hybridoma technique, this is a technique to produce monoclonal antibodies. So more of this sa next topic pa siya. So I'm not sure if familiar mo, but you have a myeloma cell and your spleen, no? Capable of producing B lymphocytes ba? So ang B lymphocytes will produce antibodies, no? So imo i-mix ang imong B lymphocytes as well as your myeloma cells. So you have an infinite line of antibodies. And then monoclonal pag siya. So it only reacts with a single antigen. Okay? So such antibody will react with the Fc portion of the human antibody attached to your RBCs. Okay? So you have two types of your antiglobulin testing. So direct and indirect. So direct gani, it is to detect in vivo sensitization of your RBCs. So this is used to detect autoimmune hemolytic anemia, hemolytic disease of the newborn, and the sensitization caused by drugs or transfusion reaction. Okay. So here, RBCs are washed, detected with your antibody or complement. So on direct, using your polyspecific antiserum. So polyspecific antiserum gani, it could be ang giuse na reagent is IgG, okay? Antibody against IgG or against any complement, C3d, C3b, C4b, and C4d. Okay, so if your RBCs are attached with those substances, so possible siya mo bind to it. So if antibody is present on the patient, your anti-human globulin is able to bridge the gap and cause agglutination. Okay? And if ever indirect gani, it's most useful in detecting your allo antibodies. Okay, so patient serum is combined with known antigen. Okay, so to detect if naaba siya ato na antibody. Okay? So the washing of your serum removes antibodies not directly attached to your RBCs. So manang important ana. Okay, so you have the procedures. I'm pretty sure naan na ni siya sa inyong immunohema. Okay? So same raman gid ilahang way sila pag-test. Ang difference is in vivo ba or in vitro inyong pag-detect. So in vivo gani is your direct Coombs test. If it's in vitro, that's your indirect Coombs test. Okay? And remember, ang Coombs is your AHG. So kaning blue. Okay? That is your Coombs reagent. Okay. So if the antibodies are already bound to your RBCs sa inyong body daan, so you are going to detect it. Katong bound na antibody, you call it your direct na Coombs test. Okay? Sige. So, para simple recall rin siya sa inyong IH. The next, you have your instrumentation. Okay, so kanang mga possible na mga automation. Okay, so you have there turbidimetry. So the principle here is that when particles combine, light scatter increases. and absorbance of the sample increases. Okay? It may also use nephelometric means to measure forward light scatter. Okay? So nephelometry gani using your forward light scatter mo niyang gi-measure. So if without using particles, the soluble antigen antibody complexes are detected at 1 to 10 micrograms per ml lang. Okay? If ever you're using particles na good, okay, particle enhanced immunoassays, then it can already reach, makadetect na siya as low as nanograms per ml. Okay? So you have the particle counting immunoassay or PASIA. It uses 1 micrometer latex particle. So it measures the number of residual non-agglutinating particles in a specimen. Okay? So the latex particles are coated with whole antibody molecules or with your Fab 2 fragments. Okay? So ang advantage of using these fragments lang is to reduce interference and not with non-specific agglutination. Okay? So, ang difference among turbidimetry and nephelometry. So, ang turbidimetry, straight lang siya, no? The detector is in line with your light source. When you say nephelometry, gani, mag 90 degrees na siya. So, ang imong detector is 90 degrees from your light source. Okay? So, ang forward light scatter ang iyang i-measure. Okay? So there is an inverse relationship that exists between your number of unagglutinated particles counted and the amount of unknown antigen in the patient's specimen. So therefore, if daghan ang remain na unagglutinated na particles, meaning lesser amount of your unknowns in your patient's serum, okay? Or sa imong patient specimen. So the measurement is made by the rate assay or endpoint assay. So a rate assay gani, it's the rate at which the unagglutinated particles decrease. So currently gini mo nakitaan ang pagka-decrease, okay, sa unagglutinated particles. While your endpoint, it measures the ending of the reaction. So it measures the amount of unagglutinated particles when the reaction ends, okay, by the end of the reaction. Okay? So kung pila na lang naibilin at the end. So for example, no, para sa siya ma-apply ang kaning PASEA. So it will detect serum proteins, therapeutic drugs, tumor markers, and certain viral antigens such as measles, herpes simplex virus, and your hepatitis B surface antigen. So next is the quality control and quality assurance. Okay? So in every subject yun, kailangan og QC o QA. Okay? So sa may QC sa IS. Okay, so possible na there is cross-reactivity with antibodies, no? Due to the epitopes na ilahang i-attach to. So possible na close lang to each other or they resemble each other. So therefore, how do you remedy it? So use of monoclonal antibody. Okay? So ang monoclonal antibody man good, unique na siya to a certain epitope and in a certain antigen as well. Okay? So rheumatoid factor, mo ni ang pinaka common no na maka-false positive. Rheumatoid factor, naapod na heterophile antibodies. Okay? So therefore, ang pag-treat na is pag-pre-absorb yung mudaan, no, with your serum and your RBCs without your test antigen. Pwede po na the RF samples are treated with 2 mercaptoethanol and pronase. So imuhang mercaptoethanol will actually destroy the disulfide bonds of your IgM. Na knowing na RF is also an IgM. Okay? So need proper storage. Make sure na dili pa expired inyong na-use. Sensitivity and specificity of the reagents, no? So possible man good na magka-cross-react. So, possible na dili siya specific ang test for it. Kanay yung gidetect na unknown sa patient. Okay? Sensitivity, possible put na dili ka detect as low as that or as high as that na concentration. So, check the label of the reagents. So, check your reagents. Okay? And no need for viable microorganisms. So, therefore, this is simple and inexpensive. Okay? And this is only used for screening. Agglutination test gani for screening na na siya. Okay? So this is important, again, for identification for pathogenic organisms na lisod i-cultivate. No? You have Francisella, Brucella, Cryptococcus neoformans, rotaviruses. Okay? Sige, this is a summary or mga possible makakause og false positive o false negative sa inyuhang agglutination. So, familiarize. Okay? Okay. So, kanino mga contamination sa glasswares, maka-false positive. Auto-agglutination, false positive. Okay? So, you have there no rheumatoid factor and your heterophile antibodies, makapa-false positive. Then you have there how to remedy, how to correct it. Okay? So, familiarize lang with this table. Okay? Okay, so that's it for my topic. If you have any questions, clarifications, don't hesitate you to message me in Teams. Sige, these are my references for this topic. So, Immunology and Serology by Turgeon and Clinical Immunology and Serology by Stevens. Okay? Okay. So, good luck. God bless sa inyuhang last weeks of this semester. So, thank you guys and bye-bye. Amping mo.